My name is Chris Arts. I'm an Associate Professor of Translational Biomaterials, and I fill this role both at the Maastricht University Medical Center, at the Orthopedic Department, but also at the Eindhoven University of Technology, at the Orthopedic Biomechanics Department.
My name is Taco Blokhuis. I'm a trauma surgeon at the Maastricht University Medical Center, and the scope of my work is mainly the treatment of bone defects and non-unions.
The I factor is a biomaterial, first of all. It is a 15 amino acid peptide sequence bound to anorganic bone material, and it is a osteoconductive material used to treat patients with bone defects. The mode of action of I factor is actually threefold. The first phase, the attract phase, recruits osteogenic precursor cells by making the P15 binding sites available for cell adhesion. In the second phase, the attach phase, these cells make a strong connection to the ABM scaffold and also interact with the type 1 collagen. And in the third phase, the activate phase, they first of all differentiate the precursor cells until full osteoblasts that are able to make extracellular matrix. And also it makes an environment next to the scaffold that facilitates bone regeneration and the bone formation in direct contact with the ABM scaffold.
The biggest difference of I factor to other materials that we have at our disposal is mainly the formation of bone. I factor is known, and it has been shown very clearly that I factor is capable of stimulating bone formation by attracting cells and differentiating cells in the direction of osteoblasts that have to do the actual formation of bone, which is a huge difference as compared to other osteoconductive materials. Most of the materials that we have at our disposal are osteoconductive only, meaning that they provide a scaffold for ingrowing newly formed bone. But I factor has an extra because it attracts cells and it makes the cells differentiate in direction of osteoblasts. We know there is a lot of evidence in spine on the formation of bone, and it is effective. It has been shown in vitro and in clinical studies that it leads to more bone formation, and at this point we're trying to translate those data into trauma.
In my personal opinion, the innovation of the I factor is, first of all, it's how it is the composition of the product, but also the level of evidence behind the product. Starting with the composition, I really like that it is peptide based, so you don't have all the associated problems like you have with the proteins, who are more complex. If you look at the fundamental evidence behind all the three factors, so both the attract, the attach and activate phase, it is all supported, individual papers, high-level citation index papers, and they actually have done a really good job to support all these individual parameters well in the literature. So I think that overall package of the innovation of the product, but also the support of the evidence behind it, makes it quite unique in this bone regeneration field.